Bismillahirrahmanirrahim. We start another practical paper and we discuss it and we see how we can do it more competently, and we can get as many marks as we can on this paper, which is a very easy paper, and anybody can get 35 plus out of 40 in this paper. As you know, this paper is for 2 hours, and you can have two variants. The time can either be paper 3-3 or 3-4, and all of you will be assigned, either you'll do the 3-3 or the 3-4, and both the practicals are nearly the same. They aren't very difficult. The difficulty level is the same, and the grade boundary is different for the different practicals, maybe a mark or two less or more, but nothing else. But basically, it is for 2 hours. As we start discussing question number one, ascorbic acid is important in the diet for maintaining health. Ascorbic acid can be found in many vegetables. You will investigate the effect of heating on the concentration of ascorbic acid in a vegetable extract. You will be carrying out a test to estimate the concentration of ascorbic acid in a vegetable extract. You are provided with the material shown in table 1.1. A is 0.1% ascorbic acid solution 50 cm cube. Then W is distilled water 100 cm cube. Then iodine solution 20 cm cube. Then star solution S. This is 20 cm cube, and U will be the unknown vegetable extract before cooking, that is 20 cm cube, and C is the vegetable extract after cooking, and this is also 20 cm cube. So just look at it, look at the volumes given to you, and you have a rough idea what you've got to do. If any solution comes into contact with your skin, wash off immediately under cold water. It is recommended that you wear suitable eye protection. To estimate the concentration of ascorbic acid in the vegetable extract, you will use iodine solution. The higher the concentration of ascorbic acid in the vegetable extract, the greater the volume of iodine solution used. So if you have 1 ascorbic acid, then the volume will be 1. If you have 5 ascorbic acid, the volume of iodine solution will be 5. So the higher the concentration of ascorbic acid, the greater the volume of iodine needed to reach the end point. So this is something which is information which is given to you. You need to remember this when you're doing the rest of the question. The end point is when the blue color remains for at least 10 seconds. Now, starting with the A part of the question, to find the volume of iodine solution needed to reach the end point, iodine solution will be added to the vegetable extract 1 drop at a time using a syringe. To practice releasing drops from a syringe, carry out step 1 to step 3. Fill a 1 cm syringe with distilled water W. Hold the syringe over an empty test tube as shown in figure 1.1 and push the plunger slowly to release 1 drop. Repeat this until you can release 1 drop at a time. So this is just to practice releasing the drop from the syringe. So they've asked you to do that 1, 2, 3. Then it says you will need to carry out serial dilution of the 0.1%. You will need to carry out a serial dilution of the 0.1% solution of ascorbic acid. So it has to be, everything has to be less than 0.1, can't be more than 0.1. Solution ascorbic acid A to reduce the concentration by half. So 0.5, 0.1, then it should be half of 0.1. What will be the half of 0.1? It will be 0.05. Half of 1 will be what? So 0.1 divided by 2 will give you the half. So it's concentration a half between each. You will need to prepare 4 concentration of ascorbic acid in addition to the 0.1% solution A. After the serial dilution is completed, you will need to have 10 centimeter cube of each concentration. This is important that they know the total volume left for you. So it is 10 centimeter. So you have 10 centimeters of each concentration. Complete figure 1.2 to show how you will prepare your serial dilution. Figure 1.2 shows the first 2 beakers you will use to make your serial dilution. You will need to draw 3 additional beakers. For each beaker, and add labeled arrows to show the volume of A transferred, the volume of distilled water added. Under each beaker, state the concentration of ascorbic acid solution. Now we've done this many times. This is in nearly every paper we have this question. So everybody should really understand how to do this part of the question. Now here you can see there is a clue here. It says 20 cm cube of 0.1% ascorbic acid solution A, in which you've added 0 W, which is water. But the important thing is that you have taken out How much? You've taken out 10 centimeter cube of the 0.1%. This was 0.1%. So you've taken out 10 cm cube of the 0.1%. Now, of course, how much will you add? You'll add 10 cm cube of water to it. And you can see there was 20 here and there's 10 left here. So this is the clue that you have to get from the first beaker. 20 initially, 10 left. So that means you've taken out 10 and then you've added 10 water to it. Now, just to show you how we're going to half the concentration. So 0.1 divided by 2 will give you an answer. Divide that by 2 will give you the next answer. So the answers were what? 0.1%, then 0.05%, then 0.025%, 0.0125%, and 0.00625%. So these were the concentrations that you had to make, and they were a total, they were 1, 2, 3, 4, 5. I hope everybody understands that. You have to read the question. If it says half, I remember another question, it said it should be reduced by 10 times. So just do 5 or 6 of these questions from different paper 3s and you will figure it out what you have to do and how you have to do it. But you have to understand it. Please don't just sort of make it, and don't keep on thinking that you know it and then you're pretending that you know it, but you don't know it. So don't fake it up. So you added 10 centimeter of W. Now you had added 10 centimeter of the 0.5%, which I'm drawing in green arrow. And to this you added 10 centimeter of W. Now you're going to again take out 10 centimeter. And you're going to add it to another beaker. So you have to make 5 beakers. I'll just draw them for you and then you can have a look at it. Now that I've completed the table, I'm not so good at putting everything all in line. So you see this is one beaker, this is the second beaker, then this is the third beaker, and this is the fourth one, and then this is the fifth one. So what have we done is that we have taken 10 centimeter cube out of one and put it into the second. This is called a serial dilution. So you have done this is that you have taken out of here and then taken it here and then put it in this, and then you've added 10 centimeter cube of water to every one of them. So you've diluted it by, you've halved it. So from 0.1%, it was 0.1% originally. From that, it went down to 0.05%. If you divide 0.1 by 2, you'll get that figure. Then it was 0.025%. If your math is very poor, I'm sorry, you'll have to find out a way to handle that, because everybody has some problem, so you have to learn to handle that. 0.0125 and then 0.00625. So these would be the 5 beakers that you have, and how you make these dilution. This is called the serial dilution. I will just roll it once again so that you can all see it, how I've done it. Maybe you can pause it here if you want to, but I can't get the whole picture in one. I hope this is clear to you all, and I hope this has been, you've understood how we took 10 centimeter cube from one beaker, put it 10 centimeter from the other, and then we added 10 centimeter of water, so it diluted it by half. So 0.025 became 0.0125, and 0.125 became 0.00625. Then it says carry out step 4 to step 17. Step 4, prepare the concentration of ascorbic solution as decided in A1 in the beakers provided. So you labeled those and you made those 5 beakers. Put 1 centimeter of S into a test tube. Okay, so here we have S. So we put in 1 centimeter cube of S. What was S? S was the star solution. So it says put 1 centimeter of S into a test tube. So we've done that. I'd added 1 centimeter cube of S into a test tube. Put 5 centimeter 0.1% solution A into the test tube. So then you add 5 cm cube. 5 cm cube of A, which is the 0.1%, which is the highest concentration. Then you shake the test tube gently to mix the contents. Put the nozzle of 1 centimeter syringe into the beaker containing iodine. So then you're going to take out the iodine. Now please remember, iodine is a brown color. So you got iodine solution and you take a syringe and you take out the iodine solution and you take out 1 centimeter syringe into the beaker containing. Pull the plunger in so that you get 1 cm of the iodine enters the syringe. So you've got the iodine here and you take it into the syringe and you fill up the syringe with 1 centimeter cube of the iodine solution. Right? Wipe off any excess iodine from the outside of the syringe with a paper towel. In step 11 to step 15, you will be finding the volume of iodine solution needed to reach the end point. Put 1 drop of iodine as shown in figure 1.1 into the mixture of S and A. So you add iodine to this. You put 1 drop of this here into this mixture of S and A. So this is the mixture of S and A in the test tube. Mix gently and observe any color change. Repeat step 11 to step 12 until a blue color appears. You may need to refill the 1 centimeter syringe with iodine as in step 8 to step 10. When the blue color appears, shake the test tube gently for 10 seconds and see if the endpoint has been reached. If the blue color disappears, then repeat step 11 to step 14 until the mixture stays blue for at least 10 seconds. This is the endpoint. Now, what you need to do is you need to do this very calmly because nobody is watching you. The video doesn't go to Cambridge. It's only your paper script which goes to Cambridge. So whether you do it or you don't do it correctly, it's immaterial, but don't become nervous and don't become agitated. If the color does not stay blue after adding 5 centimeter of iodine solution, stop adding iodine solution. So you needed to fill up the syringe at least 5 times because every time you took 1 cm cube of the iodine. Where was that written? You may need to refill the 1 centimeter syringe with iodine. So if you still added 5 cm cube and so then you all had to do was write more than 5.0. Then repeat step 5 to step 16 for each of the concentrations of ascorbic acid solution prepared in step 4, where the beakers where you made the serial dilutions, this one. So for each of these dilutions, then you repeat step 5 to step 16 for each of the concentration you did in step 4. So repeat step 5 to step 16 for each of the concentrations of ascorbic acid prepared in step 4. Now record your results in an appropriate table. Now this is the challenging part and this is the one which is going to get you. So there are 4 marks for this table. But please be sure to get this table. Now how do we do this table? There's always one little rule that we follow. Is that you write the independent variable in the first column. Now, what was the independent variable? That was the percentage concentration of ascorbic acid. Percentage concentration, percentage concentration, ascorbic acid. And what was the unit? Well, I've written that percentage concentration. So percentage ascorbic acid concentration should come first, right? So you draw a line here. And then for the heading for the dependent was what? Volume of iodine. Volume of iodine. So the volume of iodine would come here and we would have what would be the unit for it would be cm cube because volume is okay. So then we complete the table. We draw a line on the sides. And now the important thing is that you do not write in any way. So it was 0.1. But there's a student who does this. Well, I'm sorry, there's going to be a -1. This is going to be a -1 because you have given the percentage here in the table. You wrote here percentage, whether you wrote that or you just said concentration of ascorbic acid and you made this percentage sign. And then you've written it again here. Well, this is where you're going to get a -1. You don't get a -1. I mean is you're not going to get the 1 mark for that. Because if you have written, it keeps on saying in the mark schemes again and again, no units in the body of the table. What does it mean, no units in the body of the table? Means that you don't write this here. You don't write cm cube here with each. This means no units in the body of the table. This is what I mean. Some of you don't understand the English. It's okay. I mean, English is not our mother tongue. So it's okay if we don't understand. It's nothing to become so proud of. People who speak English very well think they're doing a great job. I'm sure people in UK or America don't speak Urdu that well as we speak English, and we are so proud of speaking English. We really confuse people. So 0.025, and so you will complete this table. Now I have just cooked up the results. Now, how did I cook up the results and why did I cook up the results like this? Because you see 0.1% is the highest concentration of ascorbic acid. So more of the iodine will be needed for that. It says volume of iodine recorded for the highest concentration greater than the lowest concentration. So more the ascorbic acid, more the iodine which is needed to get the end point. So I've just, made any because I've kept it less than 5. But it doesn't matter actually. Whatever your figures, maybe somebody else writes something else, but the basic answer was that for 0.1%, the volume should have been more, and for 0.00625 volume should have been less. Now, anybody could have written something else. I mean, the student could write this 1.2, 1.0, 0.8, 0.7, 0.6. Well, that would also be correct. So this person's answer would also be correct, and this person's answer would also be correct. So this is the confidence that you get, is that you don't have to worry about what actually the volume was. You can just cook it up and write anything, or you can just do 0.1 and 0.0625, and rest of it you can just make it up yourself. So this is how you get the 4 marks for this table. Now for labeling the axis, you got 1 mark. For percentage concentration of ascorbic acid, you got 1 mark. Then if you wrote volume of iodine and cm cube, you got a 2nd mark. Then if you give me results for all the concentration and to at least 1 decimal place, even if you said 3, you said 3.0, so 1 decimal place at least. And volume of iodine recorded for the highest is greater than the lowest concentration of ascorbic acid. So you can even do this without actually touching any apparatus. You can do this without handling any apparatus. Then it says describe one significant source of error when carrying out steps 8 to step 17. So go back onto steps 8 to step 17. Now this error is difficult to judge the endpoint. Sometimes it's blue, sometimes you think it's not blue. Is it blue? Is it not blue? It's a very light blue. And it's difficult to read the syringe as the iodine solution is too dark. So these were the only 2 errors, and there's only 1 mark for it. So difficult to judge the endpoint and difficult to read the syringe as the iodine solution is too dark. So difficult to judge the endpoint and difficult to read the syringe as iodine solution is too dark. You will now estimate the concentration of ascorbic acid in vegetable extracts U and C. Repeat step 5 to step 15 with U instead of A. Record the volume of iodine solution to reach the endpoint. Repeat step 5 to step 15 with C instead of A. Record the volume of iodine needed to reach the endpoint. If the color does not stay blue after adding 5, record as more than 5. Record the volume. Now I've recorded it is 3 cm cube and 2.6 cm cube. Now, you could do anything. I'm not saying this is the correct answer. You could do anything. Now, basically, U has got to be more than C. Why? But C, you've cooked it, so the ascorbic acid is lost when you cook it. So the ascorbic acid decreases when you cook the vegetable extract. So record the volume for U and C, and U is greater than C. That was the important thing that you had to think. Now, it says, now we go on to the next part. It says complete figure 1.3 to show the positions of each of the percentage concentrations of ascorbic acid recorded in A2. Now, which one was A2? Here, this is A2. Now, what you understand is that you said use your results. So I've recorded it this the whole line was 0 to 0.1. So half of it here would be 0.05. Then you divide this line, from here to here into half, and this would be 0.025. And then you divide this remaining portion into half, and it will be 0.0125, and then half of that would be 0.0625. You can do it with a ruler and do it very exactly. Now use your results in A2 and A4 to estimate the concentration of ascorbic acid in U and C. Show these estimates on figure 1.3 by placing the letters U and C in the correct positions along the line. Now that was very easy because you see what I did was I figured this out from here. 3 was 0.05 and 2.8 was 0.02. These were the two that I picked up. So 3 and 2.8. So U was 3 and C was 2.6. So then you have to place it in between here. So 2.6 was here. Sorry, 2.6 was here. This is not the one. Now you place this on the line. So 2.6 was 0.0125 and 3 was 0.05. So this would be U and this would be C. Now you do it here. So 0.05 was U and 0.0125 was C. Now see how I placed it on this line. So describe how you could modify this procedure. Now it says describe part 7. Describe how you could modify this procedure to obtain a more accurate estimate of the concentration of ascorbic acid in the vegetable extract U and C. Do not include the use of colorimeter in your answer. Narrower range of concentrations. Repeat and calculate a mean. Use a micropipette. So how do you improve it? Because you will have narrower range of concentration, so then you can get a much more exact estimate. So narrower range of concentrations, repeat and calculate a mean. That's usually an improvement that we can always do, and use a micropipette. So 3 points. Number 1 was narrower range of concentrations. Number 2, repeat and calculate a mean and use a micropipette. Any one of these got you the 1 mark. U is a vegetable extract before heating and C is the same vegetable extract after heating. Suggest an explanation for the effect of heating on the concentration of ascorbic acid in the vegetable extract. Now it depends. If your C was less, then means heating breaks down the ascorbic acid. But if it was the same, then you have to say heating has no effect on the ascorbic acid. So basically it is from your answer that we will calculate it. We will look at your answer. If your answer was the same for U and C, then of course we will say heating has no effect on the ascorbic acid. But if your U was more and C was less, then heating breaks down the ascorbic acid. So heating, this is not denatured. This ascorbic acid is not a protein. So heating breaks down ascorbic acid. Don't say denatured because that's only for proteins. Ascorbic acid is not a protein, it's an acid. But if the value for U and C was the same, then you said heating has no effect on ascorbic acid. Then, of course, it says in the investigation you've carried out, vegetable extract C was heated for 60 minutes. Outline how you could investigate the effect of different heating times on the concentration of ascorbic acid in the vegetable extract. So, of course, here you would use the same concentration, same temperature, you would use a thermostatically controlled water bath, 5 different times and times evenly spaced. So one you would boil for say 20 minutes, then another for 30 minutes, then another for 40 minutes, and then another for 50 minutes. So 5 different times so that you can see whether the boiling time has any effect on the concentration of ascorbic acid which remains. But you have to use the same concentration. Here you would not change the concentration of ascorbic acid. You would use one concentration, 0.1% or 1% or 2%, whatever you wanted to use of the ascorbic acid. Same concentration of ascorbic acid, same temperature, use a thermostatically controlled water bath to maintain the temperature, but of course you heat it for different times, 5 different times. So you heat it one for 10 minutes, the other for 20 minutes, the other for 30 minutes, 40 minutes, and 50 minutes. So because basically they had the 60-minute thing. So you would use different times, less than that or more than that also would be correct. B part of the question, some chemicals such as ascorbic acid have antimicrobial properties. The scientists carried out an investigation to determine the effect of ascorbic acid on the growth of a species of Bacterium B subtilis. The growth of bacteria was investigated by measuring the mass of the bacteria when grown on agar containing different concentrations of ascorbic acid. All other variables were kept constant. The results are shown in table. Now, this is always the first column. This always goes on the x-axis. Please remember that. This is the independent variable. So the first column is always the independent variable, and you remember that when you are making the table in the first part of the question. So this will go on the x-axis and this will go on the y-axis. So you will write the whole heading, the whole heading you will write this here, and you will write this whole heading ascorbic acid slash millimoles on the x-axis. You label the axis and you decide the scale. The scale, of course, is easy to decide because you can see the smallest number and the largest number on the x-axis and the smallest and the largest on the y-axis. Scale on the x-axis was 10 millimoles to 2 centimeter. You label it every 2 centimeter. Y-axis was 2 milligram to 2 centimeter. And then you correct plotting of all the 5 points and then you join them through all the points. Points. 2.5 was 9.7. Might be a little off, so I can't do it on this very accurately. 10 was 7.2, 20 was 4.7, and 30 was 3.1, and 40 was 2.4. Now you plotted them, you got 1 mark, or if you correctly plot it, and then of course you connect the lines, which is the most important thing, is that you connect it very carefully so that each one goes through the points. I might not get it very correct. You can do it with a ruler as well. Now this one of course is not very correct. So, and it must be a fine line, it should not be a thick line, and you must do it with a fine sharpened pencil so that you get it correct and you get your 2 marks for this. So this was 4 marks and everybody must try and get 4 out of 4 in this. And the last part of the question was use your graph to calculate the percentage decrease in the mass of the bacteria between 10 millimoles and 35 millimoles of ascorbic acid. Show your working. Percentage decrease. What is the formula for percentage decrease? Now it says you have to calculate. So for 10 millimoles, it was 7.2 milligram, and for 35, it was 2.8 milligram, whatever it is from your graph. So 7.2 minus 2.8 is 4.4. 4.4 divided by 7.2, the original, into 100 is equal to 61%. How do you calculate the percentage decrease? Calculate the percentage. Step 1. Find the difference. Then 20 divided by 80, which is the starting. So divide by the starting value, 0.25, and then you multiply it to 100, so 25% decrease. You must know this formula for the percentage decrease. Or you can use this value, final value minus the initial value, and over the initial value into 100. So this is the decrease percentage formula which everybody must know, and I hope you can figure it out and you can do it. That finishes this video on question number 1, and I hope this is going to help you in the final exams, which are just a few months away. Thank you once again.